Thank you very much. I'm glad to be here. We heard today, I think in the opening talk by Dave Tillman, that the challenge to agriculture looks like that we need to as much as double productivity by the middle of this century. But we also heard that average yields aren't going that direction, that the actual rate of increase of crop productivity is actually declining. And for some of our crops, the high-end crops, it looks like the yield may have plateaued. And so what I'd like to do is just try to provide a rationale that in order to address some of these questions, we really need to think about increasing yield potential, and that increasing the light conversion efficiency of photosynthesis is an opportunity to try to do that. Pretty sure I'm pushing, oh, there you go. And so many of you may have seen this equation before. And so this is often referred to as the yield equation. And what it tells you is that yield potential, and let me define yield potential. And so yield potential is the yield that you'll get out of a crop that's when the crop is grown in an area for which it's well adapted, it has adequate nutrients, adequate water, pests and pathogens are controlled as in lodging. And so you can think of it really as the genetic potential of that crop. And so the genetic potential is constrained by the total solar energy that's available during the growing season. So that's the total amount of energy that's available to the crop. And then that's worked on by three genetically determined things in the crop: the efficiency with which the crop intercepts the radiation, the efficiency with which photosynthesis converts that energy of radiation into biomass, and then the decision the crop makes of how much of that biomass it's going to partition into the portion of the crop that we care about, so the rice grain or the wheat grain. And so why are yields stagnating? And why are yield increases stagnating? And I think part of that reason has to do with that yield potential is stagnating because those things that drove the Green Revolution. In the Green Revolution, we saw this doubling of productivity on a land area basis over about a 40-year period. And what happened during the Green Revolution is this term of interception efficiency was vastly increased. So breeders got very good at selecting for plants that put out leaves and covered the soil very quickly. And then what we all know about is this harvest index, or this partitioning efficiency, went way up. And so now harvest index partition efficiency is at about 60%. And so 60% of the above ground dry matter is already partitioned into the soybean or into the rice crop. And so we think both this and this are very close to their theoretical maximum. So for best performing crops, they intercept about 80% of the radiation, and I've already said about 60% of it's partitioned into the grain. Photosynthetic conversion efficiency didn't increase at all during the Green Revolution. And the reason probably is that all C3 plants do photosynthesis pretty much the same way. All C4 plants do photosynthesis pretty much the same way. And there just isn't genetic variability for breeders to have selected on. But in the case of photosynthesis, there is room for improvement. We know that the photosynthetic efficiency in high input agriculture in farmers, say in the US Corn Belt, is about 1%. Under current environmental conditions, we think that the theoretical maximum is about 5%. And so there is the possibility there of increasing yield potential if we can increase the efficiency of photosynthesis. And so demonstrations that if we increase photosynthetic efficiency, it does translate into yield. There's various kinds of evidence for that. I think the most intuitive are the so-called FACE experiments. So these are experiments in which we can increase the CO2 concentration in the atmosphere. And so we have a FACE facility at the University of Illinois. And so this is an experiment we've been doing over the last few seasons with cassava. And so these cassava grow the entire season at about 620 parts per million CO2. Out here in the soybean field, it's about it's ambient at about 420 parts per million. And so for these cassava, we're seeing about a 25% increase in end-of-season yield. We've done this experiment with soybean over a period of 15 years. We see an average over that time of about 16%. And these rice and wheat data are from other FACE sites rice in China and Japan, and wheat was done in Maricopa many years ago. And so what you can see is when photosynthetic efficiency is increased by increasing CO2 concentration, and that depresses the oxygenation reaction and photorespiration, which is very expensive, we do see increases in yield. And so some years ago, we put together this table, and it's various ways that we think that it's feasible to increase photosynthetic efficiency. And we've heard about some of these earlier today. There's one up here of increasing the photoprotective state, and so that's been demonstrated as working. There's work going on converting C3 rice into C4 rice. But what I'd like to give you a proof of concept that it is possible to engineer increased photosynthetic efficiency. I'd really like to focus on this issue of the so-called photorespiratory bypass. And so photorespiration starts with the oxygenation reaction that's catalyzed by rubisco. And so this enzyme rubisco, most abundant protein on Earth, what we'd like it to do is we'd like it to take this CO2 molecule, attach it to a 5-carbon sugar, and make 2 of these 3-carbon sugars phosphoglycerate. But under current atmospheric conditions, about every 4th catalytic turnover, it does an anti-photosynthesis, and it fixes a CO2 molecule. It attaches it to this 5-carbon sugar, makes 1 of these molecules of phosphoglycerate and 1 of these molecules of glycolate. And so glycolate is a 2-carbon molecule, therefore it can't be recycled back into the 3C cycle, and it's phototoxic to several of the photosynthetic enzymes. And so plants had to deal with it. And the reason that this probably evolved this way is that rubisco evolved about 3.5 billion years ago when there was no oxygen in the atmosphere. And so what plants did was to evolve a very intricate and very energetically expensive way to recycle this glycolate. So they move it out of the chloroplast, into the peroxisome, into the mitochondria. It releases the CO2, it releases the ammonia. And for the purposes here, this is what it costs. For 8 oxygenation reactions, 3.5 ATPs and 2 NADPH equivalents. That's equivalent in a soybean field to about 35% of all the ATP and NADPH use in the plant. And so it's potentially it is a huge yield drag. And so what we decided to do was to introduce into the chloroplast a way to metabolize glycolate in a way that was energetically less expensive. And so we put in, back. And so we put in this synthetic pathway. And so we took glycolate dehydrogenase from Chlamydomonas. We put it in the chloroplast, and so that's going to convert glycolate into galaxolate. We took malate synthase from pumpkin seed, overexpressed that in the chloroplast to take glycolate to malate, and then it has enzymes to decarboxylate that. And so it's using less energy, and it's releasing the CO2 directly in the chloroplast, where it now can compete with oxygen and actually lower the number of oxygenation reactions. The other thing we wanted to do was, now that we've put this more energetically efficient pathway into the chloroplast, we wanted to prevent flow into the native pathway, and so we knocked out genetically this pathway. And so what this just shows is that the transgenes that we put in are being expressed. Each one of these is a different independent transgenic event. And then we did immunoblots to show that the proteins were going where we expected them to, and that our RNAi was working. And so here's this transporter, and you can see that when we use the RNAi module, we've completely knocked it out, or knocked it down 80%. And so this work was done in tobacco. It was done in tobacco because it's easy to work with genomically, but it's also a crop that we can put in the field, and it mimics a crop plant. And so we've done 2 years of field studies, and what you can see is we can measure the increased efficiency in photosynthesis. And so you can see here that the quantum yield of photosynthesis, which is a measure of efficiency, is increased by about 20%. And if we jump down here to yield, you can see that for the total plant, we're seeing yield increases of about 25%. And so this is a case study and a proof of concept that if we do increase and engineer an increase in photosynthetic efficiency, it does come through at least in dry matter above ground. And so, of course, the next step is that we would like to move this into food crops and see if it increases seed yield as well. And so just some take-homes. So agriculture does face a challenge if we really have to double yields. We believe that an increase in yield potential is one of the things that has to play a significant role. And we think really the only opportunity for significant increases in yield potential in our well-developed crops is to increase the photosynthetic efficiency. And then finally, I've given you one example, but there are a range of examples that are available to engineer these increases in photosynthetic efficiency. And so I will stop there. Thanks very much.
A clarifying question for Don? David?
Very impressive, Don. I wanted two questions, quick ones. So could you combine your approach with the non-photochemical quenching approach of the Steve Long lab?
Yes,
and get huge increases? And second question would be, can you keep the harvest index the same so that if you were on in a seed-bearing crop, that the additional photosynthate would be partitioned into the seed?
Yeah, and so the answer to the first question is yes. And so we have those plants now. We've been doing it by crossing, and we're doing the back crossing now to segregate them out. The answer to the second question is we don't know. In the elevated CO2 experiments, we do not see any drop in harvest index, so we're optimistic that it won't. And in tobacco, the harvest index is about 0.2, and our APHIS permit to grow these outside doesn't allow us to let them go to seed, and so we just don't know the answer to that at this point. But we're putting these into seed crop and food crops right now, and so hopefully this time next year we'll have some answers for you.
Cool.